delivered a TED talk. Yeah.
What? Oh, a TED talk. Sorry.
Yeah.
I thought you said a TikTok.
No.
No. Okay. Thank God. So we're live, Shiva.
Yes.
Okay. What? Oh, a TED talk. Sorry. I thought you said a TikTok. Oh, no. Okay. Thank God. Is that my voice? So we're live, Shiva.
Yes, we're live, but the problem is that we're live directly to our channel. So
I thought you said a TikTok.
Ugh.
Okay. Thank God. Is that my voice? So we're live, Shiva.
Yes, we're live, but the problem is that we're live directly to our channel. So
Ugh.
yes, we're live, but the problem is that we're live directly to our channel. So no problem. I send that shareable link to everybody, and okay, just give me 2 minutes. No problem. I send that shareable link to everybody, and okay, just give me 2 minutes. No problem. I send that shareable link to everybody, and okay, just give me 2 minutes. Okay. No problem. I send that shareable link to everybody, and okay. So I think we are ready. Dr. Molla, if you are ready, then should I use your home screen or I put my screen? I think your screen is good. So your main screen can be shared with us. Yes. So I think, yes. Okay. Dr. Molla. Okay. Then we are start to broadcast. Okay. Should I use your home screen or I put my screen? I think your screen is good. So okay, attendees are coming. Main screen can be shared with us. Yes. Yes. Okay. Okay. Then we are start to broadcast. Okay. Okay, attendees are coming.
Shubhabruto,
Yes.
I think your recorded voice is playing again and again.
Yes,
So I think
actually I put on the YouTube for a little bit, but I will stop it.
Yeah.
We have 501 attendees, so it's full.
Yeah. So go ahead, I think. Shubhabruto,
Yes.
I think your recorded voice is playing again.
Yes, actually I put on the YouTube for a little bit, but I will stop it. We have 501 attendees, so it's full.
Yeah.
Yes. Okay, should I start? Yes. Okay. Hello, welcome to Bioingene webinar. Today we have with us a young scientist from NRRI, Cuttack, India. He will deliver a talk on a very current topic, how is CRISPR-Cas accelerating crop improvement. We have received more than 4,700 registration for today's webinar. Thank you all for your interest in our webinars. I have with my team who will help me conduct this webinar, Liberty, Jacinta, Neha, and Shoma. Introduce Liberty Galvin from UC Davis, California. She will introduce our speaker. After the webinar talk, we are accepting questions related to today's topic from our viewers, from YouTube and from Zoom also. Important information like certificate collection process and password will be provided at the end of the question answer round by Shoma. Let's begin the webinar. I request Liberty to introduce our speaker. Liberty.
Thank you, Shavo. Welcome to today's webinar organized by Bioingene. Bioingene is a nonprofit organization created to promote plant research worldwide. The webinar series was designed to build a platform from where plant scientists can present their research to the world. We hope future scientists can gain perspective and inspiration by listening to the esteemed plant researchers talk about their scientific accomplishments and their thoughts on the future of plant science. We are grateful that many renowned scientists have accepted our invitation to share their research insights with us. We are also thankful to the audience for their interest in these webinars. We have received a mammoth response of more than 4.5 thousand registrations for today's webinar. You can view us through Zoom webinars and our YouTube live channel. Video recordings of today's event will be available on our YouTube channel. It is our request to all viewers to please hold all questions for the question and answer round at the end of the presentation. You can type in your question in the comment box, which will be opened right after the presentation. The certificate application link and password will also be available after the presentation. The topic for today's webinar is How is CRISPR-Cas accelerating crop improvement? Presented by Dr. K.A. Molla. Dr. Molla is a faculty scientist in biotechnology at the ICAR National Rice Research Institute in India. He obtained his MSc and PhD degrees from the University of Calcutta. He is a recipient of the prestigious Fulbright Fellowship. Dr. Molla has done his postdoctoral research at the Pennsylvania State University in the United States. from 2017 to 2019. He is interested in precise genome editing in crop plants and uses CRISPR-Cas and other advanced editing techniques for crop improvement. Dr. Molla is an expert in plant genetic engineering, transgenic technology, and genome editing. He has bagged many prestigious awards, including the Young Scientist Award by the Indian Science Congress Association, Best PhD Thesis Award by the ICAR, Early Career Research Award by the Department of Science and Technology, presented by the Government of India. He is also the recipient of the American Association for the Advancement of Sciences, AAAS, Washington for Excellence in Science. He also won the 11th Postdoc Research Exhibition Award for the Pennsylvania State University in the US. Dr. Molla has delivered invited lectures at several universities in the USA, including University of California, Berkeley, University of California, Riverside, University of Minnesota, University of Hawaii, Manoa, Pennsylvania State, and many others. Dr. Molla is currently serving as the editorial board member of Frontiers in Genome Editing and PLOS ONE Journal. I would now like to request Dr. Molla to take over and start his talk. Please, sir, you're already sharing your screen, so at any time you may begin.
Okay, thank you, Liberty, and for the nice introduction, and Jacinta, Shubho, Neha, and Shoma, all the team members. So it's really my pleasure to present and talk something about CRISPR-Cas and how it is impacting crop improvement worldwide and why plant scientists are crazy about this technology. It's a new tools. So I would share all those things, how it is impacting. And then let me begin my presentation with this slide published in Transgenic Research recently. So we have a lot of problem now in front of us, that one of the problem and the base of the problem is the growing population. So we are expected to reach 9.8 billion by 2050. And because of this growing population, we would battle for water, and we would battle for the arable land. Also, another very threatening challenge to us is climate change, because the climate change is severely impacting agricultural production by many means. So because increasing average global temperature causes the changes in the dynamics of pest and pathogens. So you can see here, the increase in the amount of global crop production lost to pest is around 10 to 15%. It was at 2016, but it would be around 20 to 25% because of this kind of aggravated climate changes. So we have a lot of things to do in order to feed the growing populations, in order to keep pace with the growing populations. So we have to, the plant breeders and the plant scientists has to improve the plant varieties that could withstand all those problems. So let me share my outline of today's presentation. So in part one, I would briefly mention that agriculture itself is not truly a natural process. So, and the modern crop, what we eat today, where from did they come? And then I would briefly explain in one slide the methods to improve our crop. So what are the methods that we have been using for a long time, and what are the current tools and methods to improve our crops? And in part two, I would go to introduce you to this exciting tools, CRISPR-Cas9, and what is CRISPR-Cas9 and how it is impacting and what can it do for crop improvement. And in part three, I would try to answer, or I would try to justify the title of today's presentation, that is CRISPR-Cas9, how it is rapidly accelerating the crop improvement. And in part four, I would brief about how are genome edited crops regulated. Until and unless we address this regulation, then the tools used for crop improvement, and we come up with very improved varieties, but until and unless we can cross the regulation barrier, we could not translate the effort in our lab to the field. Okay, so I would also try to briefly discuss that point. So, what I said, agriculture is not truly a natural process. You'll be understanding from these slides and next one or two slides. So, the first, according to paleobotanists and all the other scientists, those who are studying the early agriculture, rise of early agriculture and the origin of agriculture, and it was 10,000 to 9,000 BC ago, in the Middle East, that is called Fertile Crescent, they harvested fast agricultural crops in the form of maybe emmer wheat or einkorn wheat or rye. And then from 9,000 to 1,000 BC, this long duration, in this long duration, all around the world, different countries have domesticated different crops. It's like in Eastern Asia, we domesticated rice, and other crops have been domesticated in other parts of the world. So this is the duration when many crops have been domesticated. So what is domestication? Domestication is nothing but which would be something suitable for growing in our small agricultural land, and which would be better in taste, which would be better to grow, which would be easy to grow. So this is called crop domestication. I would go into details a bit later. And then comes this great experiment from Gregor Johann Mendel. And from 1856 to 1863, he studied 28,000 peas plant, and he actually performed the experiment on plant hybridization. He published his paper, and later that became the laws of inheritance, like laws of segregation and laws of independent assortment. And he discussed in his paper that one invisible factors is actually playing important role in that inheritance. So what we now recently know as a genes, that has been called by Mendel as an invisible factors. So we'd go detail into genes and later in my slides. So that was one of the breakthrough, how actually when you hybridize two plant and then the next generation comes, and how the trait becomes inherited from one generation to another generation, that became a little bit clear from that experiment. Now, next leapfrog in the crop improvement program is the Green Revolution that has happened in between 1940s to 1960s. And this is the photo of Norman Borlaug with wheat. So this Green Revolution is nothing but the development of semi-dwarf high-yielding cultivars for wheat and rice. So, if the plants are very tall, so they are susceptible to lodging. So they bend towards the soil, and then the panicle and the cross become mixed with the soil, and a lot of problem comes during harvesting. So it causes severe losses in the yield. So that problem has been solved by developing the semi-dwarf cultivars in rice and wheat. So, when plant actually uses their energy to grow taller and taller, and if you make them semi-dwarf, then the energy is converted to make more grains instead of making it more taller. So that was the basis of the Green Revolution, how the semi-dwarf rice or semi-dwarf wheat actually yielded far, far better than the traditional varieties. So now in 1953, in what happened in the Green Revolution, that was due to the inheritance of a favorable characters, and that increased the yield. But we did not know that it is because of gene and all those other things. But in 1953, after the discovery of DNA double helix by Watson and Crick, this is a DNA double helix model, and this is a photo of Watson. So I met him in Cold Spring Herbal Laboratory. So he was a very nice guy. So after this DNA double helix, we go into details of molecular biology and how DNA and its components actually regulating the expression of gene and ultimately regulating a phenotype. So it's now becoming more and more precise. So if you see this slide, it would be clear to you that all the agricultural produce that we eat today is not due to natural evolution. it's due to the intervention made by plant scientists and farmers, So if you see this, the middle figure is from Brassica oleracea, a common wild mustard. And when farmers or scientists have selected for terminal bud, it gives rise to cabbage. And when they have selected flower cluster, then it gives rise to cauliflower. And like when you select for stem and flower, then it gives rise to broccoli. And same like kohlrabi versus sprout and kale, when you select for different parts of that Brassica oleracea, and that actually you desired for that character. So you select for the character from generation after generation, and then you come up with the with this kind of different kind of vegetables that we eat today. So another very remarkable example is the selection for developing today's maize. So if you see, this is the age-old teosinte that has very few numbers of kernel in one cob. So now if you see modern corn, it has huge, even 1,000-fold more number of kernel in one single cob. So this is because of human intervention. It's not that it is happened naturally. So agriculture and agricultural produce that we eat today, most of them are because of human interventions and artificial selection. So now if I want to go in details how those phenotypes are actually are controlled by DNA, controlled by the invisible factors that has been described by Mendel, that we know as a gene now. So if you see any eukaryotic organism like plant, rice, wheat, barley, rye, and even in human body, all the higher eukaryotes, they are made up of trillion of cells. And they are the mass of trillion of cells. So if you see, this is an example of plant cell. And this plant cell has a nucleus, which is one of the main functionary body of this cell. So in that nucleus, chromosome is present in that nucleus. And this chromosome is nothing but is made up of a long DNA thread. So this DNA thread, this long DNA thread is the genetic material of most of the organisms, most of the higher eukaryotes. Now the few part of the DNA are functional and the other parts are not functional according to the current knowledge. So the functional unit of a DNA is called a gene, and that gene actually expresses a character by producing a protein. So this functional unit governs the character in a given organism. So that functional unit is called gene. So if you simply want to define gene is a functional unit of DNA. Now it is clear to us. So what makes the gene and what makes the DNA? If you see in details in the DNA, it is a sequence of 4 different letters that's called nucleotide base. That is adenine, guanine, cytosine, and thymine. So they make their different kind of sequences make genetic code, and that genetic code actually governs different characters in a given organism. So now, if you see the Green Revolution, I have told you, and we have seen that Green Revolution was a conversion from a long varieties to a semi-dwarf varieties, and that yielded more than their taller varieties. So now it was visible through phenotype, and that was done by phenotypic selection. So now later, when molecular biology improved a lot, then we could understand that because this shift from a lodging sensitive to a lodging resistant variety, that is taller to semi-dwarf, was because of a gene that is called semi-dwarf gene SD1. So this lodging sensitive has a fully functional SD1 gene, whether the semi-dwarf one has a truncated SD1 gene. So this loss of function is actually causing the reduction in height of that plant. Similarly, in case of wheat also, in Green Revolution varieties, this RHTB1B and RHTD1B, that is RHT means reduced height gene. So that gene played the important role in developing semi-dwarf wheat varieties like the SD1 in rice. So we are at a place to select our variety or go for artificial selection based on phenotypic characteristics. So now we know what actually causes that phenotype. So here, the 2 classical examples is here. One is SD1, another is RH1 gene in case of wheat. So if you see how crop breeding methods is actually moving towards precision and more precise it is becoming. So if you see, we are solely dependent on crossbreeding, and it takes 8 to 10 years to develop a variety. So if you want to develop a disease-resistant variety, so you have to cross two varieties. One is the elite variety that you want to make it resistant, and another is a donor variety. This is the donor variety that has the resistance power. So you have to cross them, and then the problem of this crossbreeding is, it has a lot of linkage drag. What is linkage drag? Suppose you want to introduce one desired character from the donor variety to the recipient variety, but that desired character is governed by a gene. So when you are transferring that character, a lot of undesired character may also come together with the desired variety. With the desired character. So it has a lot of linkage drag problems. So you want to bring one important character, suppose submergence tolerance. The classical example of developing a submergence tolerant rice variety. So submergence tolerance comes with brown furrow inhibitor gene, so that the husk color become changed, because it will never be segregated out the two controlling genes, because they are, submergence locus and the undesired character, they are so tightly linked that you cannot separate them. So, and it is also very labor intensive, this method. So you go to 5 to 7 cycles of backcross to purify the background, but still there is the problem of linkage drag. So you could have the problem of undesired character in your elite variety. So now comes mutation breeding. So mutation breeding is a random procedure of mutating gene or mutating DNA. So you apply to your plant some kind of mutagens, like physical mutagens, like radiation, all the radioactives, and then chemical mutagens like EMAs, ethyl methane sulfonate, all the chemical mutagens, and then you apply them in a whole. It's not at all a precise thing, and then you select for the desired character. Suppose you developed 50,000 population of a mutant population that has been developed from radiation mutagenesis, and now you have to screen for phenotype, what kind of phenotype you are looking for. Then you select that plant, and then you go for repeated backcrossing to purify that one, and then you develop a variety. So, but this has a problem. Same, screening is expensive and then labor intensive, and the mutation is random. You don't know in what other genes have been also modified due to that radiation, along with your desired character modifying mutation. Okay, so this is a gamma garden that has been in use after World War II. So this the central part is the source of radiation, and those are the plants that have been that are intended to treat with the radiation. And then you take out those plants and collect seed and grow next generation, and then you see a lot of variation because of the several kind of damages happened in their DNA, and then you select for the desired one. So this is mutation breeding. This is so random, but these two are not regulated. Okay, so next comes the transgenic breeding, or that we all popularly know as a GMOs, genetically modified organism. So it has unique advantage over the other two earlier methods. The advantage is that you could use the whole gene pool. You are not restricted to the gene pool of the same species. Like, here, if you want to hybridize two wheat plant, so hybridization and mutation breeding can generate variation from the wheat gene pool only. So you cannot hybridize a rice with wheat, or a mango with banana. Okay, so it cannot be done. But in case of transgenic breeding, you could use the whole gene pool, because even you could use a mango gene to introduce into your rice genome for a desired characteristic features that you want to develop. Now, but it has its own problem. That the problem is the integration loci is uncontrollable. So if you when you insert foreign DNA or foreign gene into your variety of interest, but where that gene is getting integrated in which part of the genome is not controllable. You cannot control that. Okay, and it has a, because of lot of fear mongering, lot of fear mongering things from the opponents, and non-science based argumentation that actually causes developing the very stringent regulatory scenario in the whole world. So it has a problem of this regulatory red tape, and also it is a high cost is involved to develop varieties using the transgenic technology. Now comes the genome editing technology, and mainly CRISPR-Cas. And this CRISPR-Cas actually can give rise to a predictable and precise variation that you want to looking for, that you want to develop. And it has the high potential to escape GM regulation, because in many countries it has already been started. They started to deregulate certain kind of genome edited crops, and you could develop a variety using CRISPR-Cas9, which would be foreign DNA free. So you don't have to have a foreign DNA inserted into the genome. So no need to keep that foreign DNA or foreign construct into that genome. So it would be a foreign DNA free one. So it gives us a lot of hope. So I would be mainly discussing this CRISPR-Cas in my later part of presentation. So now coming to part 2, and now I would introduce you to this exciting tool, CRISPR-Cas9, and how these tools is being used worldwide in different sectors to develop precise variation and to improve crop in a very rapid pace, which would not have been possible earlier with earlier technologies that I discussed. So if I want to just represent the CRISPR-Cas9 to you in a very layman language. So it has two systems. One is customizable RNA, and another is a protein that's called Cas9. So they're like this customizable RNA, you can develop a design RNA. These RNA actually, the RNA that would be homologous to the sequence that you want to change, to the DNA that you want to change. So this is the address, genomic address, the address where you want a change in your genome. And this Cas9 is like a, if I want to give you an analogy, this Cas9 is like a postman. So it has two scissors. One is called RubC and another is HNH. These two are nuclease domain of this Cas9. So this postman has two scissors, just keep in mind. And another, this customizable RNA is the address that you are giving to the postman. So postman takes that address with it, and it goes to the genome that you want to modify. And then it go and it searches for the homology of the RNA sequence with the genome sequence. So when the RNA sequence exactly match, a 20 base pair RNA sequence exactly match with the genome sequence, then the Cas9 or the postman gets a signal that this is the address where I need to make a cut, because it has two scissors, I told you. Now, after it finds this match, it binds perfectly with the genomic DNA, and then it makes a double strand break in the targeted gene or targeted DNA with its two scissors. Okay. So now, whenever you make a cut in your targeted gene, then the gene becomes non-functional in most of the cases. Now, what happens once you break or once you create a double strand break in the genome? What happens to the genome? What happens to the DNA? You know, DNA is very, very crucial part of any cell, any cellular organism. So whenever there is a break or damage in DNA, the cell takes it very seriously, and it immediately starts to repair it. So how to repair the two broken, and they want to rejoin perfectly to make the one which was originally there. So there are two repair mechanisms in cellular system. One is NHEJ, another is HDR. But the NHEJ is a predominant method of repair in any eukaryotic higher eukaryotes. So this NHJ is also a little bit error prone. So when they repair the two broken end, they randomly insert or delete new nucleotide in that cut point. So you see this red color is a random insertion or deletion of 1 or 2 letters of the DNA, the nucleotide of the DNA. And because of this new insertion or deletion of 1 or 2 letters from the gene's sequence is gone. So the open reading frame of the gene is altered. So this gene would not produce the same protein as it should produce in the original condition. Now this gene is non-functional. So this gene is knocked out. So you disabled the gene because of creating a targeted break and the cellular repair system. Okay. Another system is homology directed repair, but that is very, very less efficient, and that happens in cellular system rarely. Normally during the time of in dividing cell or during meiotic recombination. So what they do, they repair the same break perfectly by seeing the sister chromatid as a donor template. So in any diploid organism, there are 2 copies of one chromosome. So suppose one gene is broken, one copy of that gene is broken. So another copy of that gene is intact. So the homology directed repair, what they do, they see the intact piece as the template, and then they copy the information into the broken piece, so making it a perfect repair. But that is very very low in efficiency. So if you want to use homology directed repair in order to repair your gene of interest, then and you want to incorporate some kind of special changes in base pairing and in base sequences, or you want to insert or delete something exactly, not the random one. So you have to give them a donor template with a homologous end flanked to your intended change. So if you see here, the blue is your change that you want to incorporate. You in that donor template, you make 2 homologous arms. And then the cell will treat this donor template if it is present in adequate amount. A cell will treat it as a template. So they will copy this information to the broken end. Okay, and then your intended change is incorporated into the genome. So that's the homology directed repair. Now, you would be clear from this video narration.
Been adapted into a powerful tool for genomic research. There are 2 components to the system, a DNA cutting protein called Cas9 and an RNA molecule known as the guide RNA. Bound together, they form a complex that can identify and cut specific sections of DNA. First, Cas9 has to locate and bind to a common sequence in the genome called a PAM. Once the PAM is bound, the guide RNA unwinds part of the double helix. The RNA strand is designed to match and bind a particular sequence in the DNA. Once it's found the correct sequence, Cas9 can cut the DNA. Its 2 nuclease domains each make a nick, leading to a double-strand break. Although the cell will try to repair this break, the fixing process is error-prone and often inadvertently introduces mutations that disable the gene. This makes CRISPR a great tool for knocking out specific genes.
Okay.
Been adapted.
Okay, so now this CRISPR-Cas9 also allows us to deactivate or disable multiple targets or multiple genes in one experiment. So you don't have to go for one by one. So you can design one single experiment and you can target as much as 8 to 10 targets in a single experiment. So how it could be done? So you see, I told you that there is a 2-component system. One is Cas9, another is the address RNA that is called guide RNA. So if you design multiple guide RNAs and give it to Cas9, so this Cas9 takes one guide RNA, Cas9 takes another guide RNA, and this Cas9 takes another guide RNA. And this complex of Cas9 and guide RNA, they go to their designated targets. Like this one goes to gene 1, and this one goes to gene 2, and this one goes to gene 3, and likewise. So you can do editing, or you can disable multiple genes in one single experiment if you have multiple targets to disable. Okay, so multiplexing is easy with this system. So now I want to just briefly tell you about these three systems. One is I explained to you that already double-strand break is possible with Cas9 and guide RNA. It has 2 Caesar, and then it has you can engineer that Cas9 to make it a single strand break. So it is called Cas9. So the wild type Cas9 has 2 Caesar, okay? It has 2 nucleus domain. So now if you engineer that Cas9, you can make it the Cas9 with a single Caesar. So it will make a single cut in one of the DNA strand. So it is called Cas9. You can make it as a dead Cas9, but disabling both the Caesar. So it is called dead Cas9. So Cas9 has 2 properties. One is binding to the exact position where the RNA actually directs, and then it makes a cut. So it has 2 properties. One is binding to the target sequence and then cutting the target sequence. So the dead Cas9 can bind to the target sequence but cannot make a cut to the target. So I'm not going details into all the CRISPR-Cas tools that is available for all kind of biotechnological applications, but yeah, I dealt with the webinar a few days ago, so you could go through the YouTube and you could listen to me for knowing all different kind of tools, but I'm not going into details today. So in part 3, now this is the part where I want to justify my title, that how it is rapidly accelerating the crop improvement. So I would give you some examples of how it is impacting crop improvement all over the world. So this took 9,000 years. Coming from teosinte to modern day corn, it took 9,000 years to develop by different kind of selection, artificial selection, to develop modern day maize to feed the growing population. Now, this CRISPR-Cas tools is amazingly accelerating the domestication. So the wild crops could be domesticated very, very fast manner by using CRISPR-Cas9. So if you say this is an example, classical example of ground cherry, and this ground cherry, if you say this is the wild type ground cherry, which has a bushy nature and which takes a lot of space to grow. And one of the domestication characteristics is to develop that plant to a very compact, organized plant. So by knocking out those genes that are responsible for domestication, because we already know the genes that are responsible for domestication by our basic studies, then you target those genes to edit, and you make in 2 or 3 generations, even in 1 generation, a compact ground cherry plant that looks like a domesticated one. So this is another case where they knocked out another gene for fruit number. Those ground cherry actually has a very less number of fruits, but you see here the fruits per 5 nodes has been significantly increased in the edited one. So you got to compact the stature of the plant, and then you increase the fruit numbers. And then if you see here, the flow rate number has been increased 5 flow rates to 7 flow rates, and then locule has also been significantly increased from 2 to 3 or 4. So those are all happening in 1 or 2 generations by the application of CRISPR-Cas9. So you don't have to wait for 9,000 years to domesticate them by artificially, phenotypically selection, generation after generation. But in this CRISPR-Cas9 approach,
Hello. Is it audible?
Yes.
I think that Dr. Molla
Dr.
lost.
Molla's.
Oh, there he is.
Okay. Okay.
Sorry.
Okay.
Yeah. Okay, so I was. Okay, it was the slide I was discussing. Okay, so that was the classical example for a rapid domestication of ground cherry into an edible one with all the domestication characters. So now, if you see, this is a conventional tomato that you look for when you go to a grocery shop. But you know, always my father, my mother, and even my grandfather, they used to complain that those tomatoes are not that tasty that we used to have in our childhood. They always complaining, you know. Why? Because scientists and farmers, they always go for selecting the fruit size, fruit number, and other characters that would give them more yield and more benefit. And they have not probably focused on the taste of that one. So it is not as tasty as it should be, or as it was in the earlier cases. So this is another extraordinary example of domestication. This is a Solanum pimpinellifolium. This is a wild tomato. It is very tasty, but the size is very small, and it grows in very small number. So if you see, there are two back-to-back paper in Nature Biotech recently that they have independently domesticated this too. They have independently domesticated this Solanum pimpinellifolium into domesticated crops. So if you see, this is the wild type. This is the wild type one, Solanum pimpinellifolium, and it is growing like bushy and taking a lot of space. But what they have, when they have mutated ASP gene, that is a gene responsible for domestication, then they got a compact plant, you know. This is a nice picture from that same paper. This is the wild type one, that inflorescence. You can see the inflorescence here. It has the number of flowers in inflorescence is very less. Whether the one which is edited with CRISPR-Cas9 has significantly higher number of flowers in a single inflorescence. So if you all see here, I told you that Solanum pimpinellifolium is very tasty, but it has very small fruit size. So if you see, the fruit size has already been increased with this CRISPR-Cas9 application in that species. So the fruit locule number and fruit weight has already been also been increased by this rapid domestication program. So now this is another extraordinary example of using CRISPR-Cas9 for humankind, and that has not been possible earlier. You know, if I talk about hybrid seeds, and many ask me, and many people ask me, okay, Kutub, why farmers need to buy every time the hybrid seeds from the company? Okay, and it is, they cannot save their seeds for the next generation, because you know, hybrid is a technology when the two parent hybridized and F1 hybrid develops. This F1 is normally, this is called heterosis or hybrid vigor. This F1 or hybrid plant is more vigorous in almost every sense than their parent. And this first generation only gives the yield advantage over the parent. But if you save the hybrid seed and for the next generation you grow them, then the character becomes segregated, and you won't get an homogeneous yield or homogeneous higher yield of that hybrid. So every time you have to, if you want to grow hybrid seed, every time you have to go for purchasing seed from the company, especially in case of rice. So there is two papers, one is in Nature and there is Nature Biotech. What they have done, they tried to develop a hybrid seeds that could be clonally produced. So theoretically, that seeds, if it comes to market, it could be saved by the farmers to generate year after year, and they don't have to go to buy every year. Okay, so what they do, if you see, this is a hybrid seed, this is a diploid hybrid, where two component, red and blue, coming from two different parents. So that's why they are hybrid and they are more vigorous than their parent. Now, in normal cases, this hybrid, when they develop two gametes, one is pollen and another is ovule, and they go for meiosis, that is called reduction division. So they develop haploid gametes N and N. But what they have done, they mutated 3 genes and they induced a phenotype that is called mitosis instead of meiosis. Mitosis instead of meiosis, that is called MIME. And this MIME phenotype, what they do, so this hybrid would not go for meiosis to develop their gamete, rather it would not, it would go through mitosis to develop their gamete. So gamete would be diploid, not normally haploid one. So the diploid gamete is produced now, and we know that rice is selved, so self-pollinated crops. So the two gamete would be fused by self-pollination, and then it would develop a tetraploid plant in normal cases. But in another intervention, in the same paper, what they have done, they mutated another gene that is called not like dad. This gene is called not like dad, or matrilineal gene. So these, when they knocked out this gene, then the male gamete, the genome of the male gamete is eliminated. So only the zygote would be formed from only one of the gamete, that is from the female gamete. So you see, the same genetic makeup of a hybrid is retained in the zygote. And if you develop a plant from that zygote, then you develop a clonal hybrid embryo and a clonal hybrid plant. So it is exactly similar as the hybrid one. So if you fine-tune this technology, it would greatly impact the poor farmers in the developing countries, and obviously in the whole world, because it is going to revolutionize agriculture if it comes to market. So this is how we could change the entire agricultural paradigm with this technology. Now, this is another example from developing an industry-oriented development. So a corn that is called waxy corn, that has been developed by DuPont Pioneer and Corteva AgriScience. So this is, if it has more amylopectin, so it becomes very waxy in nature. And this waxy corn is very important in industry for processed food and making adhesive and high gloss paper. So if you see starch biosynthetic biosynthetic pathway, so the sucrose comes here to ADP glucose, and ADP glucose go into two directions. One is amylose, another is amylopectin. So now if you just inhibit the gene which is synthesizing amylose, then sorry for the interruption.
When you have to work from home, sometimes you can't control what happens.
Hello, I'm sorry, somebody is working in our rooftop, so in order to reach him, to stop him, so I have to stop here for 10 minutes. So I think it is not viable to stop presentation here. So I'm just continuing. He would be stopping soon, I think. Okay. Okay, so what I was discussing about this waxy corn, this is very valuable for these industrial applications. So if you inhibit the amylose production pathway, then you channelize the ADP glucose towards the amylopectin synthesizing pathway. So then you get a lot of amylopectin. So they developed these corn with more than 97% of amylopectin by deleting the WOX gene that is responsible for amylosynthesis. This is another development. This is, if you see a mushroom, if you cut a mushroom and if you keep it for longer time, that this mushroom, the tendency of that mushroom to become brown and black, okay? And if it becomes brown and black and nobody likes it and nobody would like to buy it. So what has been done by Pennsylvania State University scientists, I was working with the same lab, in Ong Yang's lab, and what they have knocked out the PPO gene, that is polyphenol oxidase gene, and this polyphenol oxidase actually causes the browning of the at the cut end by oxidizing the secreted phenolics. So when they knocked out this PPO gene, then they could able to achieve to develop a mushroom which would not brown as fast as the original one. So this is called non-browning mushroom by applying the CRISPR-Cas9, and it's very rapid. So this is another very interesting example in case of reducing or eliminating a health problem due to ingestion of some food. So if you eat wheat, barley, and rye, many Western country people, they have this problem of celiac disease and they are intolerant to gluten. So, if you go to any meeting, so I always used to see this is gluten-free bread, this is gluten-free, all these kind of things. So, because some people, if they eat that food from wheat, barley, or rye, they have some problem in their intestine. If you see what happens to them, so this is a normal small intestine, and this villi is responsible for absorbing nutrition from the food that we intake. And now, if you continuously eat this gluten, with this wheat, actually this wheat contains a protein that's called gluten protein, and this is made up of gliadin and glutenin. This gliadin actually is some small intestine of some people. They recognize this gliadin as an antigenic one. So they develop autoimmune response because of ingestion of this gliadin. One second. Okay, I was telling that this gliadin is actually recognized by immune system as a foreign body, as an antigen. So they develop antibody against this gliadin. So because of this autoimmune response, the villi became destroyed, and it looks like this kind of destroyed villi. And when you have a destroyed villi, then you won't be able to absorb nutrition as you do in normal condition. So this is when a person develop the celiac disease, and this is because of the problem of gluten. So now the problem is, the question is, do we have a tool to selectively remove or selectively disable this gliadin forming gene? Hello. Yeah, I'm back. I'm sorry to the audience for this inconvenience. I directed someone to look after this one. Okay. Okay, so this is an example of developing a low gluten wheat with CRISPR-Cas9. So if you see the gliadin gene, that is actually causing the problem for this gluten allergy. So this is the red highlighted one, which is the portion that is producing an epitope of that antigen. The gliadin, some portion of that gliadin protein is actually recognized as an antigen, and that is called epitope. So this epitope is formed from this sequence. So what they have done, they designed this guide RNA and to disable that portion especially, and they developed low gluten wheat by using this CRISPR-Cas9. So if you see this graph, this is multi-top graph. This is the gliadin fragment, extracted gliadin signal, whereas this one, blue and this yellow peak, is the one from the edited wheat by CRISPR-Cas9. So you can see greatly reduce the percentage of gliadin in the edited plant. And if you see the phenotype of this one is the control one, which is not treated with CRISPR-Cas9, and this one is the edited one, which is developed low gluten CRISPR-Cas9 mutant. So this looks exactly similar as the original one, but it has less gluten than the wild type one. So this is a great example how CRISPR is impacting to remove the anti-nutrient factor from our crop. So this is another similar kind of example of caffeine. The problem is caffeine. So we manually do decaffeinating our coffee. But if you could develop a plant which would produce decaffeinated coffee, that would be great, okay? And that is possible because of CRISPR-Cas9. So this is a caffeine forming or caffeine synthesizing pathway, and they're targeted. This is Tropic Biosciences working from UK. They are working on this one, and they have knocked out, and they're trying to knock out with that the theobromine synthase gene to inhibit the synthesis of caffeine in the coffee plant. So we would soon have a coffee plant which is naturally decaffeinated. So you would save a lot of money. You don't have to go for decaffeinating your coffee after harvest. Okay. So this is really humanitarian work is going on in UC Berkeley. Innovative Genomics Institute is the institute from cassava is the staple food for over 800 million people all over the world, and it is the major source of calories for roughly 40% of African people, and they are mostly poor. And because they don't process it, and a lot of they don't have all the resources to process it properly, then they eat very raw kind of things, and they don't follow all the proper procedure. So these cassava actually contains a toxin, cyanogenic glycosides, and that's toxic to human. If you consume regularly, means year after year, and with very less protein containing diet. So they are very poor, and they only depend on cassava, and they are eating it raw and not properly processed, and don't have any protein in support. So they develop irreversible paralysis in their mainly in children and women. So this disease is called Konzo, and this Konzo, in order to remove this Konzo from the African nations, Innovative Genomics Institute, Dr. Jessica Lyons and this Myung Jae So, they are working on this one to knock out 2 genes that are actually responsible or that catalyzes the formation of that cyanogenic glycosides. So if we have a cassava who does not have that toxin, we could save a lot of life and from suffering from Konzo. So this is under development. If I want to give you a snapshot of how it is impacting in crop protection, this is one slide. So a lot of pathogen affects our plant, and some plants are susceptible and some plants are resistant. because some plants are more vulnerable to a disease, to a pathogen, to a pest, because the plant has susceptibility genes. Actually, those genes is plant's own gene, but they help in pathogen establishment, and they modulate the host defense, and they also help in pathogen sustenance by giving them food and all this kind of stuff. This is called susceptibility genes. So now, if you see a classical example of the interaction between bacterial blight pathogen, that is Xanthomonas oryzae, and the rice plant interaction, what they do, this Xanthomonas bacterial blight pathogen, they secrete a protein that called tail protein. And this tail protein goes and bind with promoter of certain genes. And that gene is a susceptibility gene that helps in establishment of pathogen. So how they do that? This tail protein, when they bind to the promoter, then they increases the expression of the gene. So here the example is a sweet gene. Sweet gene is nothing but a transporter. So it's a protein. This gene produces a protein that transports sugar from nuclei, sugar from a cell to the apoplastic region, intercellular space. So now, when this transporter is produced more by the influence of the bacteria, because bacteria is influencing the more expression of sweet gene, then the sugar is transported more in the apoplastic region, and the bacteria reside in the apoplastic region, and they have more food. They're enjoying more sugar, which is produced from the plant. So they are tricking the plant's own system to have more food. So this is a classical example. And because we know the basic biology, then we could interrupt specifically the promoter of that gene by CRISPR-Cas9 to inhibit the binding of tail protein with the promoter. So if you say, these are the two results from Nature Biotech. One is CRISPR-Cas9, another is another genome editing technology, Talent, and these are the results here. So when they changes the binding sequences of the promoter specifically, then the tail would no longer be able to bind with the promoter. So the sweet gene would not be expressed in a higher amount. So the bacteria would not be benefited. Okay, so this is the strategy how CRISPR-Cas9 is being used for developing pathogen-resistant crop. This is an example from rice, but a lot of other things like citrus, canker, and also powdery mildew resistant wheat, like by deactivating MLO genes and all those kind of things. So I'm not going into details because I don't have time to accommodate all those details here, but you could, if you are interested, you could go through my other lectures. Now, another question. Till now, we have learned how CRISPR-Cas9 can disable a sequence, can disable a DNA, can disable a gene by introducing random insertion and deletion, which is not in our hand what insertion and what deletion would be there. But here is an example from a plant. You see, this is a Japonica rice variety, which has a BSRK gene. It's called broad spectrum resistance Cuttack gene. And that gene has a single nucleotide polymorphism. If it has a C in a position, C letter, or cytosine, then it is more susceptible to a blast pathogen. And when it has a T letter in the same position, and other sequence of the gene is same, only a single letter is changed to T, then it becomes resistant. So this is a great example of how one single nucleotide can change the appearance and can change the response to a foreign invader like pathogen. Okay, so this is how C and T difference makes huge difference in disease susceptibility and resistance. Now the question I want to ask you, is there a way to rewrite genetic letters precisely that you want to change from C to T? The answer is yes, only after the invention of this base editing tool. There are two kinds of base editing tool. One is cytosine base editor, that can cause the C to T mutation, that can change the C letter to a T letter. Another is A to G. You can make with adenine base editor that can change an adenine to guanine. So probably you remember from my earlier slide that I introduced you to Cas9 that has one caesar instead of two caesar, okay? So this base editor has been developed with Cas9, that is Cas9, and fused with deaminase enzyme. So I'm not going into details how they do all this kind of stuff, not into the basic technology, how they move, but you have to go through my other presentation to understand better how they actually changes selectively one base to another base. So this is an example from my work at Penn State. So we targeted this WSL5 gene, and we wanted to see whether we could also change a single nucleotide in a very precise manner to another nucleotide that we want to change. So we know from our literature that in this WSL5 locus, if this A is changed to a G, then it develops a phenotype which is easily detectable, white striped in the leaf. The leaf is normally green, but if you change this single A to a G, then it changes its color to a white striped leaf. So we tried with that one with adenine base editor. We developed adenine base editor for plant system, for rice especially, and we targeted this A, and you can see this is amazingly, we could convert this targeted A to a G nucleotide specifically. You see, this is a chromatogram, and this chromatogram means green peak designate the A, and black is G, blue is C, and red is T. So you can see this green peak has been converted to a black peak. That means A has been converted to G specifically by the application of adenine base editor. But you can see the other peaks, they all remain unchanged. They have not been changed. So I can specifically, in a targeted fashion, change one single nucleotide to another type of single nucleotide by these tools. And you can see this is the altered characteristics, altered phenotypes that we achieved in the mutant. So it is clearly visible that single nucleotide change can cause this kind of phenotype, and we can achieve genotypically similar kind of change with this base editing tool. Similar with our second target, and we have targeted more other genes, but I'm just giving you these two examples. Here, it is called zebra3 locus or Z3 locus. Here, we target this A, and similarly, we could convert this A in a targeted fashion to a G in the mutant plant, and others are not changed. Okay, and here we have seen similar kind of phenotypes that is expected due to the single nucleotide change, and we have achieved it with the in a multiplex fashion. In a single experiment, we did this two targeting, and we achieved both kind of phenotype. So this is now coming to an application-oriented application of base editing. So in crop production, how it is helping, 30% of the what we invest in agriculture, 30% goes to manage weed, especially in developing countries like India and all those Asian countries. Most of them, they do manual weeding. So those labor, they comes and they pluck weed from the field by their own hand, and a small instrument also they use. So this is manual weeding, but almost 30% in global scenario, 30% is gone to this weed management. But this base editing could develop a herbicide resistant plant development. So if it is herbicide resistant, so you could go for chemical weeding, so the weed would be killed, but the plant would not be killed because it is resistant to that chemical. So here, this is a target CGC. If the C is changed to a T, then the amino acid alanine is changed to a valine, and this could be done with cytosine base editor because C to T change. Okay, and these single amino acid change in ALS gene, acetolactate synthase gene, could give you IMZ resistant plant development. So this is one example. Another, this has been used with adenine base editor, and they targeted another gene that is called OSACC. This ACC gene is actually target of another herbicide. When you apply herbicide, it goes and kill a protein. So that protein is very essential for the plant's own survival. So how it kills the plant, okay? Now, if you apply that chemical and the plant dies, And if the plant becomes resistant to that chemical, then you develop a herbicide tolerant plant. So here, what they used adenine base editor to change this cysteine to arginine by changing A to G in the opposite strand, it is T to C, and they could develop a herbicide tolerant rice plant applying adenine base editor. So here's another, this is another fascinating area in plant science, and only 2 papers are available, but in other cases it is being used. This is called directed evolution. So you could change the sequences of a target gene, and you could enrich the sequence, and you could evolve artificially a gene of interest if you want, and you could develop novel kind of mutations, and it could give rise to a novel character that you never imagined. So this is a classical example from a group from Saudi Arabia. This is a chemical. This is called splicing. Splicing is RNA splicing. That's intron, exon. So in normal eukaryotic RNA, the intron is spliced out and exon retains, and then it codes for protein. Okay. So this is normal cases. For it is a very fundamental procedure of any biological system, means any eukaryotic systems. Now, if you see, this SF3B1 is a gene that is a very important component in a spliceosome or splicing machinery. Okay. Now, what they have done, this group, they designed a lot of different targets. These are all different guide RNAs. Okay. So they designed different targets all over the gene sequence, and they made a library of that construct, and they transformed. It means they made cut at several positions. Okay. And then they go for direct screening. So you get a mutant population of that gene, targeted mutant population. It's not that gamma radiation, the whole genome is mutated. It's not like that. It's a targeted mutation, directed. That's why it is called directed. So directed mutant population of that gene. And then you select for a splicing inhibitor chemical, that is GEX1A. And in presence of that chemical, what they have seen, that one kind of mutant is surviving at a very high percentage of that chemical, but other, including the wild type, that could not survive in that chemical. So what is the outcome of that study? You see, this splicing inhibitor chemical now comes as a new herbicide that was not known to the plant society. And this herbicide could be used, and this herbicide tolerant rice plant could be used to better manage the weed, because if you would not die because they have the resistance power. Okay. Hello, am I okay? Yeah, okay, good. Now, this is another example of using dual base editor. So I talked about cytosine base editor and then adenine base editor. Now, what they have done, they developed a new base editor which has both the properties, so it could perform C to T and also A to G editing by a single construct. So they have also targeted another gene that is called ACC. And this ACC gene, when they have in a similar fashion using the dual base editor, they have developed a lot of mutant population, and then this population, and they sprayed with the known herbicides, and they have seen some plants are surviving. And when they have genotyped that plant, they have seen novel mutations is giving resistance to that herbicide. So it is improving the basic understanding of how herbicide resistance develop, and also you could come out with new novel kind of herbicide tolerant plant that could be used for better weed management. Okay. Now this is almost the last part of my presentation. So how machine learning is a fascinating area. Everybody talks about artificial intelligence, machine learning. This machine learning has a lot of application in CRISPR field, because we are, I was a part of that team in Inong's lab for predicting CRISPR-Cas9 induced mutation for precise editing. So I told you that CRISPR-Cas9 normally makes a double strand break, and then the repair system develops random mutation, random insertion and deletion, so that it is the gene is non-functional. But now people are telling that CRISPR-Cas9 outcome could also be predicted. So what kind of insertion, what kind of deletion would be there could also be predicted by applying machine learning. So what we would do that, if we have a big repair data, so we could feed that repair data with our experimental details, what we have used during our experimentation, and we could develop a machine learning. And from that huge amount of data, they could develop a model, and that model would predict which guide is giving you the best percentage of knockout mutation, and which guide is more efficient, and which guide is going to produce what kind of mutation. Those could be predicted by using this predictor models. So it is still in the process and pain step. So let me give you a simple example by here. This is a 20 base pair target guide RNA, and if the fourth position from the PAM sequence is T, and if you apply Cas9, so you have a high chance of insertion of another T. So this is our prediction says. And we have seen in 2 or 3 of our experiment that if I use this kind of guide, we have maybe 40% of time we get another T inserted. So this is called prediction using machine learning. So this is another example just a few weeks ago came. So it says machine learning, they have utilized machine learning to predict the outcome of base editing, base editing. Right? Base editing means adenine base editor and cytosine base editor. Okay, so what kind of single base editing you are going to get? This would give rise if you use these machine learning tools. So now this is the last part, and one of the most important part when you talk about genome editing, you have to consider discussing crop, how it is being regulated, how it would be regulated, how people would perceive it, and if people is the ultimate user of this technology. We scientists develop in lab, but the perception is totally dependent on people. So it is a highly relevant matter. So, if I want to give you a snapshot of how it is, how the global community is actually seeing it. So they divided the genome editing technologies, derived crops or organism into 3 different types. One is SDN1, another is SDN2, and another is SDN3. This is in SDN1 that I mostly discussed, that when you use a Cas9, then you make a cut and the random insertion deletion causes the knockout. So it is random insertion deletion is 1 base pair, 2 base pair, maybe 2 to 3 base pair. Okay, so here you can see this extra insertion and deletion. So this is treated as, this is called as SDN1, and SDN2 and SDN3 are the 2 classes when you go for homology directed repair. I told you there are 2 pathways. One is NHEJ, another is homology directed repair. So homology directed repair, if you want to go for, you have to supply a donor template from the outside. Okay, so if you use a donor template, then it would be classified as SDN2 and SDN3. And now this SDN2 and SDN3, SDN3 is if you are knocking in a large fragment of gene, then it is called SDN3. If you're changing few base pair using a donor template, then it is SDN2. But mostly SDN3 is regarded as GMOs by most of the regulatory bodies around the world, but SDN1 and SDN2, they are most likely to be escaped from the regulatory red tape of GMOs. So if you see the available policies, USA, Canada, Argentina, Brazil, and those all countries, including European Union, they all have already decided about the policies, how to regulate genome edited crops. But the policies in development in our countries like India, China, Philippines, Japan, and others. But if you see the scenario, in case of, if I go back to this slide, so all other countries have a very positive note towards deregulating GE crops from SDN1 and SDN2, except the European Union. And I know India has already drafted a proposal of regulatory proposal, and they got public comment on it, and they are in process of developing a regulatory rules for genome edited crops. So if you say gene-edited CRISPR mushroom escapes US regulation, the mushroom I was talking about, the non-browning mushroom, USDA and other regulatory bodies in USA, they are saying that it is not at all would be regulated because it does not contain any foreign insertion. It is only few base pair changes in the PPO genes. Okay. So if you see, Australian gene editing rules adopt middle ground. Australia also is deregulating SDN1, but the other is regulated. So likewise, Japan also, they decided in 2019 that gene edited foods are safe, and Japanese panel also recommend for deregulating gene edited crops, but they have not come out with a policy, concrete policy till now. But most of them are positive, but in contrast to European Union. These European Union, what they say that organisms obtained by mutagenesis are GMOs and are in principle subject to obligations laid down by the GMO directive. So European Union is negative towards gene edited crops, and this has been highly criticized by all over the world scientists, because when they say that the product which is coming from gamma radiation of random mutagenesis, which has high likelihood of getting the different portions of the whole genome is mutated, they are not regulating it. But when you are mutating a targeted precise mutation in one of your single gene, then it is regarded or regulated. So it is highly some kind of from a scientific perspective, it is highly surprising. So if you see how regulatory frameworks, how regulatory bodies actually decide. So there are two kind of regulation. One is process-based, another is product-based. So process-based mean they go for the techniques, what has been used to develop the product. Okay. If recombinant DNA technology used, recombinant DNA technology means if cloning is involved, the variety would be regulated as a GMO. And it is the case of European Union, India, and China. But if you see in other countries like USA, Canada, Argentina, and other Latin American countries, they go for product-based regulation. They see the product which has been developed, they see the inherent risk of the final product, and that is the sole thing they consider. And they also go for analyzing potential risk of the new threat to the public and environment. So the example country is US and Canada. If the final product does not contain any foreign gene or foreign DNA inserted, so then it is free of any GMO regulations. But that is not the case for India and China. So now the question is how to obtain transgene-free edited crops. Here are some examples. So integration of construct into the genome. So if you see, if your Cas9 here, Cas9 is integrated, suppose in chromosome 1. And now you designed an RNA, guide RNA, and your target is present in chromosome 7. Now, the Cas9 will produce DNA, the Cas9 will produce protein here, and take the RNA, guide RNA and Cas9 complex, and they go to the target chromosome 7. And then they make a cut and the mutation happens, okay? So now, when the target is mutated, now the Cas9 is not needed, is no longer needed to be present in the genome. So in the next generation, you could segregate out you could select only for the mutant, not for the Cas9. In normal sexual segregation, the Cas9 would be segregated out and you select only for the mutant one. So you have the mutant plant and no Cas9 inserted. Okay, so this is called sexual segregation. Now you could achieve the same as transgene DNA delivery. So if you are not going to integrate them into the genome, but you express the DNA construct for a while, means until it makes a cut and causes the mutation for a very short period of time. So you are not going to integrate the DNA construct into the genome, then this is called transgene DNA expression. And before it is integrated, the DNA would be degraded. DNA means the Cas9 construct would be degraded. So your work is done because the time you have given, in that time the Cas9 produces protein and it causes the mutation. Then the Cas9 construct is degraded. So this is called transient one. So you don't have any problem with integration. Now you could go for RNP mediated transformation, that is called ribonucleoprotein. So you design, you isolate or you purify the Cas9 protein, and you also in vitro transcribe the RNA, the guide RNA, and you mix them and you deliver them inside your cell. So there is no DNA you are inserting into the cell. So you are inserting into the cell some protein and some RNA. So we know that protein and RNA would not be integrated into the cell in normal cases. So this is in this case, you would not have any kind of integration of any foreign gene. Another is also you could achieve transgene delivery of Cas9 and RNA by using viral vectors, because in normal cases, viral vectors typically do not integrate into the plant genome. So the mutated plants will not be transgenic. So now this is the conclusion of my presentation. So for the part one that I talked about, the genome editing has unprecedented ability to generate targeted and specific mutations in almost every loci. And you could do knockout and other kind of point mutation like A to G or C to T, and you could develop rapid targeted genetic diversity by using this technology. This simple, versatile, and efficient tools are revolutionizing precision plant breeding and biological studies. And it is rapidly accelerating the crop improvement. And it could be a boon for agricultural production and food security. Like I told you, the example of cassava, rice, and wheat, all those examples. And now in part three, during the regulation, what I should say, the most of the countries deregulated genome edited crops till now, except the European Union. But you know, biotechnology, familiarity, and perceptions of safety would drive the acceptance. So we have to actually what Bioingene is doing as a great job to communicate with the wider audience about the technology, the real knowledge, the real scientific knowledge. So I appreciate the effort from Bioingene. So we have to educate people with the real knowledge, not the unscientific argument, you know. So we should be more careful about what happened with GMOs, because a lot of people just said that, oh, GMOs is very bad and all these kind of things. And I discussed you about mutation breeding, how dangerous it is to treat the whole plant with the gamma radiation, and then you go for selection and you get a plant, and that plant might have been mutated in several other portions. You don't know because that has never been analyzed. It goes directly to commercialization. Okay. And now if you isolate a single gene which has been proved for a longer time, which have been studied from 50 years, that this gene is safe to use, and then when you insert that gene into your crop, that becomes a GMO, and how it is causing fear to us, I don't know. And if you come to genome edited crops, it is more precise when you got to change one or single nucleotide that is already present there. So it is not at all also a foreign insertion in some cases. So it should not be treated as it has been done with GMOs. So we have to communicate well with the people. So to know more, you could go for my few publications. So these are all things. And if you want to apply in your experiment, so this method book is coming soon. There you would get to know what are the protocols you have to follow to apply in this technology to your crop of interest. You could read this blog in ArtGen. You could find me there. You could go for, if you want to know more about base editing, you could go for this exhaustive review in Trends in Biotech. And then we wrote a forum article about this predicting stuff, predicting CRISPR-Cas9 induced mutations by machine learning. So you could go to that article if you want to design a better experiment for your upcoming study. So I acknowledge my postdoc supervisor, Professor Yinong Yang and Penn State, and also obviously Indian Council of Agricultural Research and National Rice Research Institute, and then the Fulbright, obviously, who has funded me for my postdoc and introducing me to a fascinating area of crop improvement. We are now applying those kind of technologies in our crop improvement program in NRRI. So we are hopeful to come out with a very good and exciting thing soon, and we'll be sharing with you, obviously. Thank you very much for your attention. And it's to you, Liberty, Jachinda, Shubho, Neha, Shoma.
Thank you, Dr. Molla. It was very informative and easy to understand talk. Now we move to our question answer round. Viewers, post their question in Zoom and YouTube. We have already collected few questions from the YouTube. And meanwhile, we have arranged a short interview with Dr. Molla. This interview will be available on our YouTube channel also. Jacinta from Canada will conduct the interview. I request Jacinta to start the interview.
Hi, thanks again, Dr. Molla, for your great presentation. It was very informative. We're going to open up the Q&A round. Perhaps you could just say a few words describing your journey so far as a plant scientist, so how you got to where you are now.
Oh, okay. So, if I start from my childhood, so I am from actually not totally a farming family, but we had farming land and we used to cultivate rice mainly because our staple food is rice. So I visited a lot of time in rice field in my childhood. A little bit knowledge about how to farm and how to transplant rice from seed bed and how to irrigate them and all those kind of things, And I started my early education in a free primary school. In India we have a great this kind of free education system as opposed to US. Please don't mind Liberty. So we have a very good system, free education system. So I started in free primary school, and then in my village there was a very nice school. There's this higher secondary school, this Joy Gram Janakinath High School, and I did my schooling from that school, and then I came to Calcutta to do my Bachelor of Science in Botany, and we call it as an honors, botany honors. So I have a combination of botany honors and then chemistry and geology. And from City College of Calcutta, which is governed by University of Calcutta, and that is 150 years old university and very well-known university in India. Then I went to, I was selected for doing my Master of Science from the university campus itself, Baligan Science College campus. And I did my MSc in botany from there. And, just what I could remember, what actually draws my attention to molecular biology, all this DNA, RNA stuff, that was one workshop that was actually funded by University Grant Commission. And this workshop was happening in our department, and few students are selected to participate from our batch. Only we two are selected, so I was lucky to be selected for that workshop. And first time I visualized DNA in UV transilluminator, and they did restriction digestion, and they used a solution to the DNA, and the DNA is divided into two parts. And that was very amazing to me. So how a protein could cut DNA, and they ligated. Then they ligated that two separated part into single fragment. That was really, really very breakthrough point in my life to draw my attention towards molecular biology. And after that workshop, actually that workshop was conducted by Dr. Momita Banerjee. She was a very nice and fellow teacher. So after that workshop, I decided to pursue my special paper. That is called a special paper in our MSc last semester. So we decided to go for biochemistry and molecular biology, And then I did PhD with a very famous rice scientist, Professor Shapan Kumar Datta. So he was with the team of Ingo Patrikas in the development of golden rice. He was working in Switzerland, Zurich. And then he came as a leader in IRRI, Philippines International Rice Research Institute. And then he later joined in our university. So, we are very attracted to him. So I got to apply to him for my PhD position, and I got it. So I carried out that PhD in his lab and got many kind of what I should call many kind of opportunities from that lab. And I learned a lot about how to develop a transgenic rice and what is the crucial points to develop for genome modification and all those kind of things. And then while I was preparing to submit my thesis, then I appeared for this agricultural research service. I know in the USA there is ERS, what is conducted by USDA, but here also we have a ERS system, agricultural research system service, agricultural research service. So through that agricultural research service, I got my first job at Delhi, our capital, this National Bureau of Plant Genetic Resources, that is NBPGR. So I worked there for, this NBPGR is known for the National Gene Bank, that's the Gene Bank of India. So I worked there for 1 year. And then as I have a great interest to work on rice modification, so I got a transfer to this institute, National Rice Research Institute, in 2015. And then I spent here for 2 years, and then I got the opportunity to go for Fulbright Fellowship. And obviously, no other technology attracted me other than the genome editing. And so I got to know by a Nature article published about the mushroom story about Inong Yang. And I contacted him. And yeah, I was lucky he accepted me in his lab. So that was my story of postdoc. And now I am back in Cuttack and I'm applying all those experience that I gained there to my to rice crop development and for my nation. Yeah. Thank you, Jacinta.
Thank you. So the next question, according to you, which sectors of plant science and research in agriculture are likely to prosper in the near future, do you think?
There are a lot of cross-industry application in agriculture, especially if you see it may not be that popular, it may not become that popular till now in India, but in those kind of developing countries, but it is highly used in developed countries like your country. Mainly use of drone in agriculture, and then if you want to go for precision agriculture, and obviously artificial intelligence to decide what part of the crop field is affected by satellite imaging. And then obviously nanotechnology, those nano formulation of fertilizer, pesticides, all those kind of things that is less harmful than the normal chemical fertilizer. And if you see another very interesting that I came across a few days ago, that is 3D printing. That 3D printing is also impacting the agri prototypes productions of different kind of parts. So it actually allow you to rapidly evaluate all different kind of designs and what design would be suitable for your application. Like if I want to give you an example, I told you about cassava. So cassava, great, you have to peel out the skin of that cassava, okay, before you eat. So you have to grate that cassava. And also they recommend, in order to remove the cyanogen, you have to grate the cassava and you have to dry it and then soak it and then drain the water, okay? So you have to grate the cassava. So if you want to design a cassava grate, then you can go for 3D printing, and you can visualize a lot of models and you can select the best one that would fit. So it is great in 3D printing. Even expert believes that. Obviously, it may sound like science fiction, but expert believes that hydrocolloids means the substrate that form gels with water. That's called hydrocolloids, okay? And these hydrocolloids could be used to replace the base ingredients of our foods, like with renewable sources like algae, duckweed. You could use those things to make as the base of our food by using this 3D printing. If you want to know, you have to go through other reading. It's not possible, then I have to go for another presentation on 3D printing, okay? So this is the area I think is very, and then if you see, if you ask me top technology that I want to mention, obviously genome editing, CRISPR-Cas9. Yeah, and another is obviously vertical farming. So if you are urban people can also do farming in a very limited space. So they can go for vertical farming. If you see the example of Israel, they're doing it in a very huge amount. So yeah, these are the areas I think.
Great, that's really interesting. I'd never thought of using 3D printing in agriculture. So thanks for that. One of the questions that I saw pop up a lot in the Zoom chat and the YouTube was, do you consider genome edited CRISPR crops as GM crops?
Okay, I think that I have already answered, but still I would briefly mention. So in the current scenario, regulatory bodies, they're dividing the gene edited crops based on how and why they have produced, like SDN1, SDN2, and SDN3. So SDN1 is the simplest one. So if you knock out a gene, simple. So most of the world regulatory bodies, they consider SDN1 as a non-GMO. Suppose if you want to get rid of the toxin from cassava, and you knocked out 2 genes from cassava, and you developed a gene-edited cassava that does not have that toxin, that cassava would not be regulated as GMOs as per the recent regulation of most of the countries. Even I read the DBT proposal, Department of Biotechnology, India, they have similarly classified SDN1, and it would not be regulated as GMOs. It was in draft proposal, but it is not final. But if you use to knock in, but I have not, I skipped that slide. I was talking with Liberty about Pam Ronald from UC Davis. So she is a very famous scientist, you know. I visited her lab also and met her postdoc, Oliver Dong. So Dong is a very good friend of mine, and they published recently a paper to integrate the whole carotenoid biosynthetic pathways, cassette, into a safe harbor locus. So, you know, if you see the genome organization, there is some portion of the genome. If they harbor foreign gene insertion, it would not hamper the total genomic scenario or total genomic construction. It would not hamper, it would not have any bad effect. That called safe harbor loci of a genome. So this technology, CRISPR-Cas9, allow you, what was the problem with normal transgenic technology? When you insert a foreign gene, it was not, we were not able to control where it is going to integrate. Now, if you use CRISPR-Cas9 to integrate a foreign gene using homology-directed repair, then you could insert your gene of interest at the point where you want. So what they have done, the Pamela Ronald has done that whole carotenoid biosynthetic pathway cassette, they inserted into a safe genome harbor. And then they developed a new variety which has golden color. So that's a vitamin A rich rice, and that should be called as golden rice. And we know that where it is integrated, now it is easy to analyze, okay? So If this kind of develop happens, then it would be regulated as GMOs. Most of the regulatory body says, because it contains the biosynthetic pathway gene inserted into the genome. Okay, so in order to produce the trait vitamin A, you have to have that cassette integrated into your genome. So this is called SDN3, and this SDN3 is GMOs. But SDN1 and SDN2 are most likely not GMOs. Yeah.
Do you think that integration being classified as genetically modified would also include if you inserted extra copies from the same genome? Take, for example, golden rice, if you inserted those carotenoid genes, which already exist in rice, in those safe harbor genes. Would that still...
If you ask my opinion,
yeah,
it should not be. It should not be. But I am not the controlling authority of every regulatory bodies. Yeah. But the problem is, if you I told means in my introductory slides, I gave you example of whole genome shuffling by normal crop breeding. The huge genome shuffling, mutation breeding, huge random mutation. A lot of different kind of things happening, but they are not being regulated. But when you know that this gene is very much okay, and the product of this gene, this protein is also okay, long history of safe use, then it is regarded as genetically modified. Small modification is regarded as genetic modification. Large modification is not regarded as genetic modification. I don't know why. Yeah, this is my scientific view. Yeah.
Okay, last question for me. Do you have any advice for today's youth who are considering a career in plant science?
Oh my God! So I'm not that big to advise people, but yeah, I have some point because here I manage some student and I guide some students, few students. What I tell to them, I could share that with the audiences here. So if I go selectively for PhD students, especially because they need the suggestions, okay, because that's the most frustrating part of anybody's life, most struggling part, PhD life. So the first point that have to keep in mind whenever you enter to your PhD program, whatever you are in your personal life, you are very less organized, you are messy in your apartment, that does not matter, but you have to be organized in the lab. Very, very organized, when to do, what to do, okay, you have to write everything what you are going to do today. So what I do still now, I write in my notebook, these are the points that I am going to accomplish today, and I tick it one by one. So this is one way of organizing yourself. So you know what you have to do and how far you achieve. When you tick 1 and 2, you feel something confident, and you feel energized. Okay, I have done this 2 work. But if you have all 5 works in your mind and you started doing one, you have another thing coming in your mind. Oh, you have that work is left, that work is left. So you are going to be distracted by your preoccupied thoughts that you have 5 things to do. So it's great to do, to make a list what you're going to do. And obviously another thing is time management. So it's like PhD, I don't know, it varies with lab to lab, but in normal PhD, in a good condition, PhD student is their own boss, or their own boss. So they have to manage their own. So they have to know, they have to prioritize how they will spend their time, when to make fun, when to go for chatting with friends, when to go for coffee, and when to work. Okay, so then there, if you go to PhD is a continuous process. So you have to continuously develop your academic skills by reading more and more. So it's not that you are going to get great ideas without reading. Whatever, you may be a great thinker, but you cannot think about a good idea without reading what is going on in the current scenario, what is the real problem, what is going on in the current current literature. Okay, so you have to read more. And another important thing, third point that I want to emphasize, the self-motivation is another thing. So you always failed in experiment, and you have to be self-motivated. Nobody is there to motivate you, and your guy did not know what is going on in your mind. You are gone. Oh, this 2 consecutive experiment failed, and you thought my PhD is not going to happen. I'm not going to get the degree. Okay, so this is the common scenario with everyone. Even it happened with me. But again, if when you go to bed and if you think in that direction that okay, this 2 has failed, these 2 experiments have failed, maybe third time I would be successful. Okay, and maybe something good is waiting for me. So, and also you have to read some story of previous PhD student, previous great scientists, how they have failed and how they have succeeded in their consecutive attempt. So those are the things I think main thing that should be kept in mind as a general thing, and other things are lots. I'm not going into details on all those things. Yeah.
Great, thanks for that. I think we're going to pass it back to Shuvo to wrap up.
Hello. Thanks, Jacinta, for doing that session. And I want to conclude that there is so many questions. Most of are related to, you have already covered that thing. I am also trying to answer a few of them, but one or few, if we want to ask you that our viewers from YouTube asks that the off-targeting Sherlock process applied in the animal system, but is it okay for plant? Can you answer this question?
Okay, so what I got from the question is Sherlock is a technique to detect the presence of any foreign DNA or any foreign nucleic acid. Means especially Sherlock has been developed by Feng Zhang lab from MIT. So this is a nucleotide detection method. So they use Cas9 to detect a specific presence of a specific nucleic acid in any sample. Suppose if you want to go for diagnosis of COVID-19, so you have to go for your nasal swab and throat swab, and then you go for testing whether the viral RNA, the COVID-19 RNA is present there. So if you apply Sherlock, then Sherlock is made up of Cas9. Cas9 is a brother protein of Cas9. So that go and find any RNA and then make a cut there. So this is a chemistry. I'm not going to details. So whatever, if you apply Sherlock and if you go for detecting the RNA, the viral RNA, if it is present in the nasal swab, you could use Sherlock to detect whether it is present or not present. If you get a positive signal, so it means you are infected with the virus. If it is negative signal, you are not infected. So this is how Sherlock works. Now, Sherlock could also be used in plant system because it does not depend on a system. It depends on extract that has nucleic acid. So are you extracting it from animal? You extracting it from a diseased plant? It is all same. So it should work for plant system. Yeah. But I don't know why the viewer asked me off target and Sherlock. I'm really confused about this. Yeah.
Okay. Thank you for that answer. And we don't have any specific questions on that because those are the easily available in review and papers, and they are very basic questions. So I conclude here that thank you, Dr. Molla, for your valuable time. Hope to see you soon on the Bioingene platform with your new inventions. Your talk and interview will be available on our YouTube channel. I am sure your talk has inspired many of our viewers. Thank you. If this is a formal end for you, if you want to stay few minutes, you can stay. But now I hand it over to the Shoma to give your viewers some important guideline regarding the process of upcoming webinar registrations and certificate collection. Over to you, Shoma.
Hello. Thank you, Dr. Molla, for the wonderful webinar presentation. I'm sure your talk has inspired many of our viewers. Thank you for teaching us about CRISPR and making it so easy to understand with your illustrative slides. Hope to see you again soon. We are thankful to the audience for attending the webinar. Hope you enjoyed it. Thank you, Shubho, Liberty, Jacinta, and Neha of Team Bioingene. We owe a special thanks to our active volunteers who have helped us put together this event. We congratulate them on their successful participation and thank them with a certificate of participation. The link for certificate collection is now open for all the registered attendees and available on our website. We have already displayed the password that you will require while filling out the certificate application form. You can also get the link and password from the comment box here. Please fill out the certificate application form carefully so that there are no spelling mistakes. Please type in the correct password. An incorrectly filled certificate application form will be automatically discarded. Also note that the certificate application link will only be active for 2 hours from now. No requests for formal or informal will be entertained after this time. The certificate of attendance for the webinar will reach you within 7 working days by email. If you are still awaiting your certificate after 7 days, please write to us. Our next webinar is on genomic assisted breeding for chickpea improvement by Dr. Bharadwaj Chellapilla, ICAR-NRRI, India, on 1st July 2020 at 11 a.m. The registration for day after tomorrow's webinar will close at midnight today. Please visit our website or Facebook page to register. The webinar is free and open to all. To learn about the process of registration and certificate application, please watch the descriptive videos on our YouTube channel. The webinar video recording will be uploaded on our YouTube channel. Please like, share, and follow our Facebook page. Please like, share, and subscribe to our YouTube channel to stay connected to Bioingene. Our YouTube channel also consists of videos about the process of registration, certificate application, and about the upcoming webinars by Bioingene for the month of July 2020. Thank you all for making this webinar successful. See you soon at our next webinar.
Hello, I want to request Dr. Molla, can you stop your screen sharing? Hello? Can you stop your screen sharing?
Sure. Is it okay?
Okay. No, not now.
It's okay?
I think now it is okay.
Mm-hmm.
Yes.
Okay.
Now,
Full screen.
I'm request Shoma to give some certificate. to those who are helped a lot during the webinar arrangement.
We cannot thank enough our volunteers. Here are some certificates to Tata Srinivasa Rama Satya Sandeep, to Agnimala Sai Teja, to Jatsi Srivarsha, to Almaru Krishna Chaitanya. Here is the password, the application link, and the website link. And you can see some of the upcoming live webinar details. The next webinar is day after tomorrow. The registration will close at midnight tonight. Please hurry if you want to register. And the registrations for all the webinars in July are open. You can register them. Thank you.
Okay. Thank you, Soma. And I want to thank all the team of Bioingene, Galvin, Neha, Jacinta, and our speaker, Dr. Kutubuddin A. Molla. We are met soon in our next webinar. And thanks to all the audience. And our certificate application link is now open, and it will be open for 2 hours. It need a password. We give it in that description box from in Zoom as well as in YouTube. So apply your certificate. We will give it within 7 to 10 working days, and see you in the next webinar. So thank you all of you. Thanks, Galvin and Jessica. It is quite different time in your country, but it's a very nice, and we got so many appreciation from the viewers for this platform, and we are able to answer few of their questions, and maybe later they can search more from the Google. If they need something, they can got from the websites. So we can't give them the presentation slide because we can't keep it for our storage, but we can provide the recording. It will be available soon in splitted video in our YouTube channel. So please subscribe to our channel for all the next live webinar. Like us in the Facebook page and inform others. Those are the work on the plant science. Thank you. Thank everybody. I'm officially close this meeting. Thank you.